Welcome to the 2023 Classes Without Quizzes program, and thank you for joining us. My name is Annie Makepeace. I have the pleasure of hosting the event today. I received my MS degree from CFANS in Applied Plant Science in 2015, and then I served on the CFANS Alumni Society board from 2016 to 2022, and I continue to serve as chair for this event, even though my time on the board has come to a close. So this program was established by the CFANS Alumni Society way back in 2002 as a way to keep alumni and friends connected to the college and to highlight new, exciting, impactful research that's all being done by our faculty. So over the last 22 years, more than 1,100 alumni and friends have joined in this learning adventure, with many in person when we used to host this on the St. Paul campus. So we've enjoyed expanding our reach, being able to get outside of the Twin Cities metro area since shifting this program to a virtual event in 2020. So this program is one way that we bring to life our board mission to foster relationships that benefit our college alumni, students, and friends by sharing the awesome and important research that's going on throughout CFANS. So we hope you enjoy the program. With that, we're excited to welcome you to Curating Curiosities in the Bell Museum Collections, where we're highlighting CFANS experts who serve in curatorial roles for the Bell Museum. We'll start with an introduction from the director of the Bell, and then we will have lightning presentations from 3 of its curators. We encourage you to add your questions to the chat throughout the presentations, and we will moderate a brief Q&A after the speakers finish all of the talks. And then at the end, you'll have the option to interact with the faculty presenters directly in a breakout room for more engagement and questions. So those rooms will be open for about 10 to 15 minutes after the program closes, and you'll be able to move from room to room if you would. And just as a quick note, housekeeping note, if you could keep your mics muted for now, just until we get to the Q&A portion, that always helps minimize distraction. And now it is my pleasure to welcome Holly Menninger, Director of the Bell Museum, to provide us with an introduction.
Myself. Hi, everybody. I am so delighted to be here. So I'm going to click over to my slides now because I have the fun job of, all right, is everybody seeing? Cool. Of introducing you to the Bell Museum. So we are so proud to be the state's Natural History Museum and Planetarium, and to be part of the University of Minnesota and CFANS. We just celebrated our 152nd birthday, I should say. That this is a slide from our recent 150th anniversary. We were actually, let's see, make sure I can, there we go. We were founded on Leap Day, 1872, and charged with collecting, preserving, and interpreting the biological record of Minnesota, and for that to be held in trust at the University of Minnesota. So that connection between the U and natural history is 152 years strong. And I thought, since especially we celebrated a Leap Day this year, we can sing happy birthday to ourselves. Collections, scientific collections, are core to who we are and what we do. And you're going to hear from some of our scientific curators in just a moment. But I wanted to give you a smattering of what do we mean by those natural history collections, these organisms that we preserve for research, for study, and for study and to inform natural resource management throughout the state and beyond. So our scientific collections include 1.2 million specimens. They include birds and mammals and fishes and amphibians and reptiles and plants and fungi and DNA, genetic resources. And we take care of and study those resources daily. So those collections are stewarded by our faculty curators. As I mentioned, you'll hear from 3 of them who have appointments in CFANS today. We have a total of 7 scientific curators and 2 collections managers who are really responsible for managing, adding to, loaning, and studying our scientific research collections. You'll hear from those folks in just a minute, but I wanted to make sure we sat back and that you understood a little bit more about all that the Bell Museum had to offer. So in 2018, we moved to a new public building on the St. Paul campus. Many of you alums from earlier decades may remember the previous location of the Bell Museum on the Minneapolis campus. We moved and opened to the public in July 2018 to great fanfare and acclaim. We're really excited by our new building. In fact, we still call it our new building because we think it's a really special place. We recently refreshed our mission and vision for the museum. And so what I'm going to do in my next few minutes is talk to you about how we live that mission to ignite and sustain curiosity for nature and the universe. And we do that because we envision a hopeful future where all are inspired by nature and empowered by science. How do we do that? Well, certainly, if you come into our public museum, which is on the corner of Larpenteur and Cleveland, right on the edge of the St. Paul campus, you'll get to enjoy our state-of-the-art exhibits. Here's a scene of our biodiversity wall, so you can see some of our scientific collections on display for our audiences. I should mention that our collections are not located in our public museum, but are in academic buildings on the St. Paul campus. And you'll get a sneak peek of what some of those spaces look like when you hear from our curators in just a couple minutes. We like to say that you can take a walking tour of the ecosystems and biomes of Minnesota in a couple of hours. And so here I have a scene from one of our world-class nature wildlife dioramas that represent real places in Minnesota and bring to life in a very immersive way the ecology of this place that we call home. Of course, we can take you into the past. And so we have an awesome woolly mammoth, a giant beaver, and other animals that lived during the Ice Age when Minnesota was covered with ice 19,000 years ago. We've added a planetarium. And so our new public facility, this is something new if you've visited the Minneapolis location that you'll see here. We have a 120-seat digital dome that creates this immersive experience where you can go to the far edges of the universe. You can touch down on the surface of Mars. We can help you zoom along a neuron and do all kinds of incredible things in this particular space. And it's been a real joy to add space science and astronomy as part of that larger story of natural history that you'll experience at our museum. We have a lot of programs. We're expecting to see about 150,000 visitors this year when we count both our K-12 students that visit on field trips. We were rocking and rolling earlier today. You could hear their sounds of the students having fun in the hallways. But then certainly with great public visitation. We host a number of public programs that allow us to showcase the science that happens in the museum, as well as for us to be a gateway to the science that's happening across the University of Minnesota. So this is a researcher in neuroscience who's leading a family of visitors through a game about that helps you understand how the neuron works. Our curators and their graduate students are huge partners for us when we are doing public engagement and connecting with visitors. Every summer, we host a tent outside where our curators and students are actually processing specimens. So it's helping people understand and see how do we create these scientific collections that are so important to research. And in fact, those folks are going to take this show on the road. This normally happens in the summer outside the museum to the Gathering Partners Conference, which will be happening in partnership with Minnesota Master Naturalists, I believe in May. I'd be remiss if I didn't talk about some of our space programming. And so I'll, before the end of today, I'll let you know about a really special event happening in April. We host star parties, both locally at our public museum, but we lead a statewide star party at sites distributed all across the state of Minnesota. We want everyone to enjoy the night sky and observe the sun. Hint, hint. More on that later. Of course, our work is not confined to our public museum. We hit the road and we have really invested and expanded our statewide engagement team so that we truly can be a museum without boundaries. And so our team is leading community STEM events in partnership with libraries, community centers, tribal organizations all across the state. And so you can see Ellie, one of our team members, showcasing some specimens at a community community science night. We also have a portable planetarium that we take on the road and are able to do star shows and set up in gyms and community centers. I'll just highlight, we are expanding our summer camps from beyond our public building. For the last couple years, we've partnered with the North Central Research and Outreach Center in Grand Rapids to host a summer camp. And this year we're expanding west and south to include the Northwest Research and Outreach Center in Crookston and the Southwest Research and Outreach Center in Lamberton. I'll be happy to share some links for folks who might be in those regions to learn more. That desire to be a museum without boundaries and that work is not just the work of the educators and science communicators that are in our public museum, but it extends to the work of our scientists and curators. And you'll hear a little bit more today about the work that Sushma and team are leading, a project to turn dead wildlife into scientific specimens so that we can better understand ecology and disease, as well as a program that we've long had at the Bell Museum where we create online access to our specimens from across Minnesota so that the public can see and engage with the biodiversity that we have here in the state. It's also a resource. It's called the Minnesota Biodiversity Atlas that's used by natural resource managers every single day. I hope my last point is I think we are an institution with incredible momentum. I'm very honored to be the new executive director at the Bell. I've had the great pleasure of being on the Bell team, on the leadership team for the last 6 years. And I can tell you, we have some really exciting things ahead of us. We're powered by an incredible team, an incredible creative team who are really thinking outside the box. So look for some exciting new things from us in the next year related to climate solutions, related to traditional ecological and astronomical knowledge, and related to the digitization of our scientific research collections. And so with that, I think I've stayed within my time. Please visit our website and our program and events calendar. I'll preview a couple things that are coming up later this month. Here's my contact information, and I am so excited to hand it off for you to hear directly from some of our faculty curators.
Thank you so much. That's so exciting, Holly. It's so cool to see everything that you guys are up to. And thanks for the introduction to the Bell and everything that it has to offer outside even of the walls of the museum itself. It's just a beautiful world-class natural history museum, and we're so lucky that it's attached to our university. So next, we look forward to hearing from Kassandra Ford. She is the Curator of Fishes and Mollusks. So thank you, Kassandra, for joining us today.
Thanks so much for having me. I am going to find my slides here. There we go. All right, so thank you for having me. I am incredibly excited to talk about my role as curator of fishes in particular. I'm also curator of mollusks, but I know a little bit less about them, but they are still an incredibly important resource for Minnesota wildlife and the researchers that are here in our home state. So I am Dr. Kassandra Ford. I am by training an ichthyologist, which means that I study fish. I'm an evolutionary biologist, which means that I like to study kind of the ancient history and the historical context of the organisms that I'm looking at. I'm also a functional morphologist, which means that I like to look at why an organism is shaped the way that it is and how that might be influencing what it eats, how it eats it, and where it's living its life. One thing I wanted to touch on is this idea of why we actually have museum curators and what that means for my career and what I'm actually doing with my life. The collection of a natural history museum is incredibly important. There can be the part that the public sees. So Holly showed some amazing pictures of what our biodiversity wall looks like at the Bell. Here's another example of the Natural History Museum in Berlin that shows off really the fish collection that they have there, with this gigantic cube full of glass that shows a whole bunch of the different fish specimens that they have in their collection. But there's also the part that the public doesn't always get to see. Here are some pictures from other museums about the shelves and the walls full of fish sitting in ethanol in jars that are used every single day for research purposes. These are the areas that the public doesn't always get a firsthand look at, but it's really the heart of the museum. It's the part that is really being used for day-to-day research, for interest, and can be brought out for some public engagement in terms of showing off what the collection has, how it's used, and why it's important to keep it up and running. Another thing that the collection can be really important for is examining the specimens that we have. So I am a user of a micro CT scanner a lot. So this is like a little mini medical CAT scanner where you can scan an organism and look at the inner workings of the fish or other organism that you're trying to study. You can look at bone, you can look at muscle, and really get an inside look at the organism without damaging any of the specimens. This can also be put onto open source networks so that we can share that information with other people and they can share it back with us so that we can do really worldwide research, kind of from our own backyard or from our own little office on campus. And the collection is huge for showing fish biodiversity. Our collection contains some of the 30,000 different species of fish that there are in the world. And these fish, honestly, each individual fish can tell us a little bit of a story. I love studying an idea called convergent evolution. This is the evolution of similar characteristics in different groups of organisms. So this could be something like the spines in hedgehogs and echidnas, or flight in birds and bats. So these completely independent groups of organisms have come to kind of a similar conclusion in terms of what they look like, how they're protecting themselves, or how they're actually moving in their environment. Some of the groups of fish that I study are electric fish from South America and Africa. Here are some heads, my talking heads that I like to discuss when I'm thinking about research. We have a South American electric fish on the left and some African electric fish on the right. And if we dive underneath the cover or the skin of the fish, we can see their skeleton. We can see their head and the bony shapes underneath it that are really the foundation of how these fish are interacting with their life and what they might be eating. So here's that again. We have some South American and some African fish. And I'm really interested in the skull because the skulls do so much for us. They help with breathing, they help with feeding, they help with mouth brooding, which is what that fish on the bottom is doing. It's not eating the little tiny fish, it's actually trying to protect them by holding them in their mouth when they're super young, little tiny fishies. We can think about skulls and we can actually make some inferences or hypotheses about what the fish are doing or what the organisms are doing based on what their skull looks like. So the skull of a lion and the skull of a cow are going to look vastly different because these organisms are interacting with their world in much different ways. The lion is a predator and trying to obtain organisms for food, whereas the cow is an herbivore and really only trying to get grasses and plants as food. And so we're going to see differences in like their overall size of the skull, the teeth, things like that, that showcase that we kind of have some differences in lifestyle and diets across these two organisms. So I take those ideas and apply it to fish skulls. Here's just kind of a smattering of a whole bunch of fish skulls that I am looking at. And if we highlight two of them, we have these kind of elephant trunk-like snouts on these two specific species of fish. And I wanted to know, since these fish look somewhat similar in their head shapes on the outside, how similar are they on the inside? So I used some math and statistics, which I will not bore you with at all, and actually found that despite the outsides of the fish looking somewhat similar, the underlying bony parts were actually far different than we expected them to be. And so while their heads might be similar in shape and they might be doing similar things in the environment, that doesn't mean that kind of the bony makeup of the fish is going to be identical. This is an example of convergent evolution, what I was talking about before, and we have a few different examples of that across these groups of electric fish from South America and Africa. those concepts are the basis of the lab that I am just starting. I'm brand new to the university as of this past fall, and I have started the Convergence Lab, where we are looking at these ideas of similarities and differences across organisms and trying to tie that to where organisms are living, what they're eating, and how they're going about their day-to-day life. And with that, I would like to acknowledge the land that I've done all of this research on, and would love to answer any questions that you have in the breakout rooms as we move forward. I don't know how I did for time there. I tried.
Yeah, I think we're ahead, so that's great.
Perfect.
Yeah, and as a reminder, feel free to put questions in the chat, and we will be monitoring that, and we will bring those questions forward after we have the rest of the speakers conclude. So feel free to pop those in there or hold on to them. You'll have a chance to ask them verbally if you'd like to as well at the end. So thank you, Kassandra, for sharing your work in using these museum collections to study these things like evolution and morphology and history. I know that fish are near and dear to many people in Minnesota, so it's really cool to hear a little bit about your research, whether that's here locally or out in other parts of the world. So 30,000 species of fish, I had no idea. That's incredible. Next, we will hear from Ken Kozak, who is the curator of Amphibians and Reptiles. So thank you, Ken, for joining us today.
Hi, everyone. See, I'm wondering if I'm sharing here the right way. I don't think so. Okay, looks good. And you're hearing already my bullfrog call launched already. So here the bullfrog stealing the show at the beginning. What a great way to start. As I'm the curator of Amphibians and Reptiles at the Bell Museum. I'm also a professor in the Department of Fisheries, Wildlife, and Conservation Biology. And today I'd like to talk to you about a new project that we have going on in the Bell, basically with students in my lab, looking at American bullfrogs in Minnesota. Probably many of you know that bullfrogs are native to Eastern North America, but they're not actually native to Minnesota. They get quite close, but they've been introduced in a couple places. I'm going to talk to you a little bit about what we know about where they are and how we're going to study about where we might know where they are and where they may be going in the future. American bullfrogs, they're native to Eastern North America. One of the reasons when they become invasive, that's what we're worried about here, is because they're one of the largest amphibians in North America, getting up to 8 inches in length, which is pretty large. They're aquatic habitat generalists, so they can exist in lots of different kinds of habitats. So if they get into a place where they're not native, they can kind of wreak havoc on local aquatic food webs and ecosystems. They're really explosive breeders. Females lay up to 20,000 eggs. Lots of them hatch in the tadpoles. They remain as tadpoles for up to a 2-year period. Those tadpoles are herbivorous, so they also interfere with basically herbivorous parts of the food web. And overall, bullfrogs can live a really, really long time in the wild. They have up an 8 to 10-year lifespan. Part of the problem with them is they can eat anything they can fit in their mouth. If you look on the bottom lower right there, that's an American bullfrog eating a koi fish out of somebody's backyard pond. Okay, so we're talking they can eat some really, really large things. So they have, we have lots of interesting fish that we're interested in here, especially game fish, so they can kind of wreak havoc on that part of the ecosystem as well. One of the problems with them also too is that if they get into a place where they haven't been before, they're highly resistant to predation. So if they get into a place where they're naive predators, they have toxic skin secretions, which the local, fauna has hasn't evolved defenses to. And they're also the way they feed is they're secretive sit and wait predators. So they don't move around a whole lot. So they're kind of, they're not very visible to predators as well. It can also make them kind of hard to detect in ecosystems at times. They're also habitat generalists. So they have a really broad thermal tolerance range, a preferred range of 15 to 32 degrees centigrade. And so they're particularly prone to inhabiting new areas, especially in North America, where it's cold in Minnesota because of these broad thermal tolerances. So now I'm showing you here now is a slide. The green here is where we have the native range of American bullfrogs, and introduced parts of the introduced range. You can see here, basically in the West and the Pacific Northwest, bullfrogs have wreaked havoc on ecosystems out there. Maybe some of you have seen that in the news. But we also have them here just north of their native range, and now, getting up pretty far here into northern Minnesota. So whether these are separate introductions, we're not sure, or whether they exist everywhere in between, we really don't know at this point. But they're considered 100 of the worst alien invasive species by the Global Invasive Species Database. So there's something we should really be taking care to look at here locally in Minnesota. They're largely introduced through aquaculture. Basically in the late 1800s and the early 1900s out in California and the Pacific Northwest. It was for frog legs, and it's been for frog legs in other parts of the world. Also through the pet trade, and probably in places like here, probably in bait markets in Minnesota. The tadpoles and things make their way in the bait markets, and that's probably how they've gotten out here, because we don't have a whole lot of bullfrog farming going on here in Minnesota. One of the big things is, as I said, they disrupt aquatic ecosystems. So they're explosive breeders, and they compete with everything because they're generalist predators. And they also compete for space in aquatic ecosystems. And another big concern about them is they act as reservoirs. So bullfrogs are somehow able to carry emerging infectious amphibian diseases, like something like chytrid and ranavirus. They act as reservoirs. And they don't actually have the negative effects of those viruses, but they take them out and they introduce those viruses into basically the native amphibian fauna. So American bullfrogs, where else are they? So this is talking about like the local part of the project here. They're presently documented in 17 counties. The sites you see here with dots these are actual specimen records. So we have these in the Bell Museum in the collection. Up here in this northern county here, these are citizen science observations. So they're not confirmed by specimens. They could be misidentifications. We don't know. But at any rate, bullfrogs have not been systematically surveyed across Minnesota. So the key questions being explored by Bell Museum herpetologists, basically my lab, is like, we're going to look at where else are they? Is their geographic distribution limited by climate? So if you notice, they kind of get native to the bottom of Minnesota, but they don't make it much further to the north, right? And in places where they've become really widespread in the Pacific Northwest, the climate is much more moderate than it is here in Minnesota. So we want to look at where else are they with some systematic surveys I'll talk to you in a little bit about. Ask, is the geographic distribution currently limited by climate, right? And also, where are they predicted to occur now, so we can use models to predict where they occur now and where they might spread in the future as the climate changes, which I'm sure you're all familiar with, given the early spring that we've had here in Minnesota. So where else are they? One of the things we're in the midst of doing right now, we're going to be starting up this spring, is we're going to document with traditional biological surveys of presence. So that would be basically visual encounters over here, and also doing larval dip netting, using nets to dip net for larvae across selected sites, drainages in Minnesota where we know they occur versus drainages to where they're adjacent. And then using breeding call surveys. So you just heard the breeding call there. So if you heard that around here, you know that you heard that foghorn kind of call. That's a frog that's not native to Minnesota. That's an American bullfrog. So another way we're going to be looking at where else are they is to use these databases. Holly mentioned these in our introduction. We have the Minnesota Biodiversity Atlas, which folks can submit observation records to, so they can report where they've found bullfrogs. And also this database, which was developed by a grad student of mine, a master's student of mine, who's since moved on to work for the Minnesota Department of Transportation Herp Mapper. So what we're going to do is use citizen observations and specimen data from museum databases like the Bell Museum database, and citizen science databases like Herp Mapper to model environmental suitability. And so with that, we can do things like ask. So this is actually, if we look here, in Minnesota, we can look at places where, well, if we were to take these localities and build a model and look at habitat suitability, we could see these are places in California where bullfrogs were introduced that match the native niche. But over here, we can see the introduced niche, or the introduced set of environmental factors where bullfrogs can exist, is actually larger than the native niche. So there may be some evolution going on there that's helping them to spread. And so that's one thing that we're going to do here, one of these things that we're going to model. We're also going to ask where the species will be in the future. So are they currently limited by their current niche, or have they expanded? And what's going to happen as the climate changes in the future? Is that even going to make them expand beyond their native niche or even their evolved niche and where they're at? Is that going to open up more environmental or climatic spaces for them to occur in? And so for this, we're going to use those same databases with collection records in them. And then what we do is we take GIS maps of basically their data on what the climate is going to be like geographically in the future. And we can put them onto a map and ask, well, where is the species suitable to occur currently? This is done with a salamander here. We haven't done this for our bullfrogs yet, but we did this with a salamander in our lab that lives in the Great Smoky Mountains National Park. And what would happen if it warms 3 degrees Celsius? So salamanders are kind of like the opposite of bullfrogs, right? If you will. They like really, really cool places, right? And so in this particular case, we could see that this salamander that is basically fairly widely and continuously distributed in Minnesota is predicted to contract its range by about 60%, actually, as you can see over there on the right. And so that's where we're at. So what are the conservation implications of this work? So it's going to tell us whether our American bullfrogs are expanding their ranges across Minnesota, right? Or are they in places where we don't know they are? It's going to tell us what environmental factors might limit their spread now and in the future. And with this information, we're going to be able to ask, is there an immediate need to mitigate the spread of bullfrogs before we end up in a situation where we might have them disrupting food webs and wreaking havoc like they are doing now in California and the Pacific Northwest. And I'd like to, stop here with the big acknowledgement that this work was funded by the Minnesota LCCMR. It's a new project that just began this year. For those of you that don't know that, with the LCCMR, it's called the Legislative Citizen Commission on Minnesota Resources. And they're really funding a lot of really interesting biological and natural resource work these days that are, helping us basically understand and solve environmental problems across Minnesota. So that's one of our new projects going on in the herpetology lab. And I'd be happy, I don't know where I'm time, but I'm happy to take future, further questions in the breakout rooms if you're interested. So thank you for listening and chiming in.
Thanks, Ken. Interesting work studying bullfrogs, their locations, evolution, future population spread, looking at the conservation implications, a lot of stuff going on there. And I already saw one question, I think, pop up that we'll address after the next speaker. So we'd like to welcome our final speaker, Sushma Reddy. She is the Curator of Birds. So thank you, Sushma, for speaking with us today.
Thank you. My name is Sushma Reddy, and I am the Curator of Birds at the Bell Museum. And today I'm going to be talking to you about a new initiative at the Bell Museum to expand the use of natural history collections to study ecosystem health. As you learned from all of the other previous presentations, natural history museums are really about collections. The Bell Museum has about 50,000 bird specimens in its collection. And like most museums, including the Bell Museum, less than 1% of the specimens are on display for the public to see in our exhibits. So you're probably asking, why? Why do we have so many and what do we do with them? And where are these? Well, you saw from Kassandra's talk that these are the collections we have in our scientific collections that are housed in the academic part of the university, not in the main part of the Bell. And we use this for research of various sorts. But before I talk to you about this new initiative, I want to kind of bring in the history of natural history museums and kind of talk about why we have so many specimens, and how we get these specimens, and what do we do with these specimens. So here's an example of one of our research drawers, our collection drawers, and you can see that we have multiple individuals of the same species in this cabinet. All of these are individuals that were collected at different times and different places. Most of these are coming from different parts of the Midwest in this case, but in other cases, we have specimens from all over the world. And again, you know, an important question we get from people from visitors is, why do we have so many, right? And so the simple and a related question that I want to make sure you understand is, why do we want more? And the simple answer is to document diversity, right? Each specimen tells a story. It represents a species found at a particular place and a particular time. And these specimens help us understand nature. They help us understand and address questions, research questions, such as, what are different species? How are they related to other species? And do these species look differently in different parts of their range? Have they changed over time? How do they use their unique features in their ecosystem to interact with other organisms or other parts of their environment? And all this we can learn from a museum specimen. Having a bird in the hand, as the old adage goes, can really help us learn a lot about its environment. How do we get these specimens? How do we end up with 50,000 specimens at the Bell Museum and continue to grow this? Well, this is often driven by scientists asking some of the same questions I mentioned before. And to research these questions on biodiversity, scientists often go out and collect specimens that they're interested in and in the areas they're interested in. So over the past 150 years of the Bell's history, and this is still continuing today, curators such as myself and other bird researchers have traveled all across the globe to study birds both near and far. And the specimens they've collected for their research are archived at the Bell Museum so that other scientists would also be able to use them for additional studies, in addition to the studies that they were initially designed for. And specimens can come to museums in other ways. For example, people often find dead animals and bring them to us. And this may be something you've experienced yourself. As you're walking down and you find a dead animal on the sidewalk, you might have wondered, what happened here and how can I help with this, right? Often these are animals that have hit a building or a vehicle. And sometimes, but not always, sometimes, and I'll tell you more about this, if these are fresh and intact specimens, they can be really valuable to science. So every day, thousands of wild animals are killed due to natural or human-related causes, such as collisions with windows, buildings, power lines, windmills. And these dead wildlife are often just discarded or overlooked, but we're trying to start this new initiative that says each of these animals contains a wealth of information. So we're organizing this community-driven effort to gather these specimens for science. We're asking people if they feel comfortable enough to pick up these freshly dead animals and bring them to us. We're also creating a new network of institutions across the state that can help us store this material so that people can bring this to an organization that's close to where they live and contribute to this effort. And to our knowledge, this is the first state to have like this statewide initiative to organize such a massive effort towards salvaging wildlife and using it towards museum collections. So what would we do with these specimens? Well, anyone who's watched forensic shows know that the dead tell tales, right? So the same is true for wildlife. We can learn a lot about animals as well as their environments by examining a body. And in these bodies, there's information that's stored about what they ate, how they moved around in their habitat, and how they interacted with other animals. And so different parts of animal carcasses are useful for tracking the spread of diseases, tracing the prevalence of toxins, assessing environmental conditions, and understanding ecological interactions with other animals and plants, such as their diet or their parasites. with the salvage wildlife specimens that we will obtain, we are going to be saving material from different body parts, such as feathers or fur, skeletons, and tissues for all different types of genetic and chemical analyses. And I've listed a few on this slide. We will be taking multiple samples of different organ types so that we can use this to really maximize the information we can get from each specimen and use it and be able to use it for future studies. Some of these we aren't even sure how they'll be used in the future, but we want to be able to preserve as much as possible to create a baseline of information that other researchers can build on when they're studying biodiversity. So all of the data, all of the materials, all of these samples will be archived at the Bell Museum and will be available for agencies, scientists, and educators in Minnesota and beyond to use for future studies. Why is this important, right? How natural history museums can contribute a lot to conservation. A lot of studies in recent times have shown that bird populations have dramatically declined. For example, in just the last 50 years, there are 3 billion less birds in North America. And we don't know the specific causes of each of these population changes, but we know that this is a trend that might continue to increase because humans are increasingly going into spaces where wildlife are. They're building into places that were normally where they normally weren't before, and the impacts of climate change is continuing to escalate. So we know that this is going to be a bigger issue, but knowing how, where, and who is at risk in terms of biodiversity can really help us to manage conservation threats. And natural history collections can play a really important role in conservation because they give us a record through time. So we can compare the new specimens we're getting through the salvage program to our historical ones to better understand how each species is changing and why, and what we can do about it to mitigate these effects. So I just told you a lot of information about this brand new project. We're hoping that as spring rolls around, people will start seeing a lot of wildlife around. And if you notice any dead animals and you want to help, we would really welcome it. So you can find out more about this program by going to our website. You can also hold your phone up to the screen and get the website link through this QR code. And if you want, you're also welcome to email us with more information. We'll be updating the website frequently to track our progress on the project, so check back often. We will be reporting the number of specimens we get, where they were found, and what we are learning from them as we go through this project. One thing to note, I put this picture of someone picking up a bird. This is, don't do it like this. This is my husband picking up a bird with his bare hands. If you do want to help, make sure that you're using gloves or a plastic bag as a glove. We want to encourage people being as safe as possible when they're picking up these birds or mammals. But you can learn all these details on our website. And you're welcome to help us study and do more science at the Bell Museum. Thank you.
Thanks, Sushma. Thanks for sharing your work and how species tell us stories about their environments and their connections to nature, and how that information can help support ecosystem health and how we can help. Now we feel like we have a job to do, which is great. And 50,000 birds in our collection, that's amazing. Again, just like I'm learning a lot today. So we've covered a lot of ground here today, everybody. Hopefully you found it valuable to hear more about the Bell and its connection to CFANS and all the work that goes on behind the scenes. So many of you, I'm sure, have visited the Bell. It's cool to hear a little bit more about the work that's going on that we don't always see. So I'm sure your interests are piqued after hearing them discuss their research. So at this point, we'll open up for questions from the audience. You guys can submit either via the chat. I think there might have been a couple that came in via the chat, or feel free to unmute and ask a question directly to one of the speakers. But I think we can start. What do we have in the chat so far? Let's see. There's a question to Ken. Will you talk about native species, especially those that are threatened?
Yeah, thanks for that question, Judy. I guess I'd like to maybe expand a little bit on the context of your question. Will I talk about native species in the research and how bullfrogs might threaten them, or maybe you could expand a little bit on the question. I could address it better.
Judy, are you still there? Do you want to clarify
You
your
may be
question?
you may be muted, Judy, if you're not realizing that, or maybe she stepped away for a minute.
If she's not there.
Like we got a fish question while we're waiting.
Sure, let's go to that. We got a question on how diverse is Minnesota fish are Minnesota fish populations compared to other states?
I think we are maybe average in terms of our level of diversity, but one thing that we are definitely strong at is our enthusiasm for protecting our fish. So between game fishes and our non-game fish, which have recently been renamed non-game instead of rough fish, I would say we're being very forefront thinking in terms of trying to make sure that our ecosystems are suitable for our fish as we're moving through climate change.
I think she maybe sent a follow-up chat, Ken. I'm curious if our native populations are declining. And just a comment that it's interesting people aren't seeing deformed frogs in recent years. Are the native populations declining, Ken?
Oh, I was just putting it in the chat here.
Oh, okay.
So, yeah.
Feel free to share.
Yeah.
Kassandra, I did have a question for you. What makes a fish electric?
Oh goodness, I knew that this was coming. So there are several groups of fish around the world that are considered electric. The probably most commonly known one is the electric eel that's found in South America. But what a lot of people don't realize is that there are about 500 other species of electric fish around the world. One of the groups, South American electric fish, includes the electric eel, but the eel is the most like powerfully electric. There are specific repurposed nerve cells or muscle cells that these organisms have evolved into an organ that creates an electric discharge that they're able to control. And most of these electric fish, if you stick your hand in the tank and grab them, you wouldn't feel it. It's a very small discharge that they use for communication and seeing around them with an electric field. So kind of like echolocation or an electromagnetic field.
Fascinating evolutionary tool, I guess. We do have one for Sushma. How does Minnesota compare with the rest of the US in terms of the decline in bird numbers? Are we experiencing more or less decline in Minnesota?
Yeah, thanks, Carol, for that question. So I have to admit that I haven't looked at the statistics very closely to compare it from state to state, but the statistic of the 3 billion less birds is across North America, and it's across all bird species. And, we've had, and in some cases, very few cases, the numbers have actually gone up, but most of them have really declined. And what's really interesting, to me, in terms of the contributions we can make from this program is to not just understand what's going on in Minnesota, because obviously we're not an isolated area, right? We have connections to the rest of North America. And so we can use the information we're getting from what other museums are doing and other institutions are doing in different parts of the country and kind of compare information to see if there are kind of higher increases or higher or more accelerated declines in some regions than others. And so we don't really have a baseline information for all this information yet. But we're hoping that once we create this program, once we get these data in, we can compare it to other places where they have big salvage programs like Chicago, which is another big flyway for migratory birds, and really compare if our numbers are different than theirs.
Good question. So there is an additional question that I might initially pose to you, Sushma, and then see if either of you, the other speakers, have additional comments. But Lisa says, I'm a crane and wetland enthusiast, and one thing I'm seeing in a lot of our beautiful parks and waterways is plastics. What are the negative impacts that you have witnessed regarding this issue? Any of you guys can answer, but since she mentioned the cranes, Sushma, is there
Sure.
anything you?
Yeah. Actually, this is one of the reasons why we want to do this project, right? We don't actually know the impact of a lot of things that we think that we just take for granted. And plastics are a perfect example. We're finding there's something called microplastics, which are the product of a lot of different things that we use plastics in that we don't even know plastics are in. They're really, really tiny. And sometimes they're formed from the degradation of larger pieces, but mostly they're in things like cosmetics and clothing. And they just get into the water system, they get into the food system, and can end up in wild animals in ways that we've never predicted before. So we're finding that seabirds that are living in areas that humans don't normally live in different parts of the world, have like microplastics in their gut. And so one thing we can do in the Salvage Wildlife project is to be able to screen for this and see, are there ecosystems where this is happening more? Are there species where this is happening more? And how is it impacting them? So just like with the classic example of DDT, where we didn't understand the impacts of pesticides until we were able to compare it to other to collections and really see what things were like before DDT and things were like after DDT, can we really see those effects? And so we're hoping to have a baseline and hoping to have more information. But we're really worried about things like plastics and microplastics in particular, because we really don't know how it's going to impact wildlife going forward. And they've also found it in fishes. Isn't that right, Cassie?
Definitely, yeah. Between microplastics and even regular plastics, because like Ken's example with the bullfrogs, fish will eat anything that fits in their mouth, and that includes plastic, because they don't know that it's not a shiny, silvery fish. So
I'm going to ask one last question of Holly.
Oh, sorry. Can I just add regarding that microplastic issue? Another really cool collaboration that Belle is involved in is with the Minneapolis-St. Paul Long-Term Ecological Research Project. And there are a team of folks that are trying to understand the effects of urban contaminants, including microplastics, on behavior, particularly of mammals, including squirrels and chipmunks and their kin. And part of that work involves a collaboration with our curator of mammalogy. So there's more. This topic is very much on the radar of our scientists and those across the university.
Great question. I have one wrap up, then we're going to move on to the last part of the program. But there's a question around what CFANS majors are a pipeline for each of you and your fields of study. If people are interested in learning more.
We are all in the Fisheries, Wildlife, and Conservation Biology department, but there are also many different majors that can lead to research as well. And I would also say that I would never discount anyone who has a different major if they have an interest in scientific research. There were people in my grad school who had music majors.
Interesting. Are the other curators also fisheries and wildlife? All of you guys? Okay.
Oh, the three of us on
the others that aren't here today.
Yeah. They're also, as faculty, we sit on multiple graduate program appointments. So we're on conservation sciences, I would say all of us for sure, and ecology, evolution, and behavior. So I recruit a lot of students. I recruit students that are interested in ecology and evolution. Those would be in the College of Biological Sciences. And then, I guess from the herbarium perspective, there would be people from plant biology and people that are interested in plant ecology as well. So we kind of get pipelines from multiple directions, from applied and basic research directions. I would say adequately covers that.
And I'll add
questions.
Sorry, just wanted to add
Nope.
one more thing, is that sometimes we also get requests from art students and people from the College of Design to come and use our specimens to be able to draw and illustrate. And also they use the specimens to think about how form and function relate. I'm losing the right word for describing that, but
Well, mimicry. Well,
yeah.
as part of that too. Yeah.
So yeah, we have interest in the collections in lots of different ways. And what we're trying to also constantly do is try to find ways of promoting the collection so people know they can use this.
Yeah, I hosted
That's awesome.
a
You guys
really
have quite
great
the reach.
artist in residence, like it was years ago now, Jeff Milliken. But he did a really interesting exhibit. It was both photography based and specimen based, and it was really cool the way he portrayed collections from an artistic perspective, and somewhat it's kind of a strange, macabre sort of way, but was entertaining for some of the stuff. It was really cool to work with people like that. And so he spent time in the bird collection, the mammal collection, and also the amphibian and reptile collection, and put things together in a really cool way.
That's awesome. I love the intersection of art and science. So it sounds like you guys are right in the middle of that, which is great. So thank you guys so much for all of our speakers taking some time to present to us today. Thank you guys for all attending. We're not wrapping up just yet. Holly has a couple of exciting comments about some upcoming events at the Bell. So I'm going to turn it back over to Holly briefly. Go ahead, Holly.
All right. Share my screen. Did you know Thursday, March 14th is Pi Day? And starting tomorrow, the Bell Museum is celebrating. So we invite you to come stop on by and help us continue a chain of the world's longest never-ending number. We'll have a cart activity in our Horizon Hall, and you can participate in a really cool visualization through the weekend. So that's one thing I'd love to invite folks to. We also have on Saturday, sorry, I'm trying to advance my screen here, is one of our is Spotlight Science, which is a monthly event we host that features researchers at the university and from state agencies and other nonprofit organizations related to a particular theme. The theme on Saturday is plant genetics. And so we'll have some researchers, some grad students, folks sharing different dimensions of their work. What's really exciting is it often involves hands-on demonstrations. We like to blend, as you heard, the art and science together. And it's a great opportunity to have conversations with grad students and senior faculty. So that will be from 10 to 2 at the Bell, largely in our Horizon Hall on Saturday, March 16th. And last but not least, I alluded to April 8th is a solar eclipse. And we are going to be open. We're typically not open on Mondays at the Bell, but we're opening up this day because it's a pretty big deal. And so we invite folks to come on out, participate. We'll have all kinds of solar activities, and we'll have opportunities for people to safely, and I want to repeat, safely view the sun. Note, Goldie has his own pair of eclipse glasses. We have those glasses on sale in our Curiosity shop right now for folks who want to get ahead of the game and get those, but we welcome you to stop by on Monday the 8th. Some parts of the country, it'll be a total eclipse. Here in Minnesota, we're expecting it to be about 75% covered, which is still pretty spectacular. So come on over.
Very cool. I'm excited for that. Now I'm going to turn the program over to Mary Bichetti. She is the Director of Alumni and Constituent Relations before we move on to the breakouts.
Hi, everyone. I just want to thank you all for joining us today. As Annie introduced at the beginning of the event, this is an Alumni Society organized activity to help alums and stakeholders get connected back to the college. Every year we feature a different topic, and I hope that you will consider joining us again next year. I also just want to give a shout out to Annie and Colin Robinson and the other members of the committee who helped plan and put together the event, and thank you to the speakers as well. And then just one thing I wanted to mention is we have a mentor program that matches students with professionals, helping them explore their careers. And that question came up about majors and careers. I invite all of you, whether you are an alum or not, to consider being a mentor or reaching out to individuals who want to support students in their college career. And then lastly, we're going to be celebrating the CFANS community at Borealis Night of Excellence on April 18th. So consider coming and joining us. With that, thank you, and back to you, Annie.
Thank you, Mary. And thank you very much for joining us. And we look forward to seeing you next year.